Let's start. Welcome, everyone.
Yeah, hello all. Welcome to our webinar. So I'm Aashish Chaudhary. I'm the Assistant Director of Business Development at Kitware. Along with me, I have David Manthey, the Technical Leader at Kitware, and both of us like to welcome you to our webinar on ImageDePHI. Slide 2, David. So first, I will give you a quick introduction to Kitware, then describe our ImageDePHI tool and its core capabilities. David Manthey will cover more details of image reduction and show us a demonstration of ImageDePHI in action. We will wrap up with this slide covering some helpful information as you take your next steps in working with ImageDePHI. Again, a reminder, if at any point during our presentation you have a question, feel free to type it in the question window for this live stream. We'll take a break for questions later on, both before and after the demo. So about Kitware, Kitware was founded in 1998 and currently has about 220 employees across the US and Europe. We primarily perform software and AI research and development services for our customers in government, industry, and academia. More people know about our popular open source platforms like CMake, VTK, ITK, and Paraview than they do about Kitware. We are proud of our long history of accomplishments in open source and open science, as it lines up well with our vision of making the world a safer, healthier, better informed place. And since 2020, we are 100% employee owned. Next slide, please. We are a highly collaborative company with research efforts primarily funded by the US federal government, involving commercial and academic partners. We provide custom development, support, and training on open source platforms. Our expertise spans healthcare and life sciences, including digital pathology, which is what we're covering today, surgical planning, medical imaging, and bioinformatics. Alongside, we also work on energy, manufacturing, defense applications, simulations, HPC, and data analysis. Deep learning is central to our work, with strong emphasis on ethical and explainable AI. Next slide, please. Some of you may already know about Kitware, but in general, most people know Kitware through the open source platforms we create and maintain. We're strongly committed to open source and open science, building vibrant communities, publishing results, and encouraging collaborations. You can find all our open source technologies at kitware.com. Today's webinar focuses on anonymizing image data for digital pathology. And again, as a reminder, please type any questions you may have during this presentation into the chat window, which we'll cover before and after the demos. Next slide. So now let's talk about HistomicsDK or ImageDePHI, the work that we are presenting in this webinar. So Kitware has been developing tools for histology and whole slide imaging for more than a decade. In this space, we're best known for HistomicsDK and the Digital Slide Archive. Together, they provide a powerful set of tools and a user-friendly interface for organizing large collection of images, managing metadata and annotations, and visualizing results from both human and AI workflows. These platforms also support automated image processing pipelines, including standards methods like nuclei detection and the latest foundation models. It's mature, reliable software designed to work seamlessly with your data wherever it resides. So before we go any further into this presentation, I would like to invite David to talk more about the core topic of this presentation, which is the image de-identification. David, take it away.
Yeah, so what we're here talking about today is sharing whole slide image data. And so we just mentioned that we have our platform called HistomicsDK, where we can look at histology data and run machine workflows on them. But you might want to use your own tools and programs to do this. But in any case, in general, you have these whole slide images, and when they come from human subjects, we have to de-identify them. So in order to do this, in order to share data so we can use it, we have to remove protected health information and personally identifiable information. There's a whole variety of reasons why you might share images. Some of this might be providing test images for algorithms, or even just feeding them through algorithms where the algorithm tools are not run where it's in a secure environment where you can have PHI. Some of it's providing exemplars of health and disease conditions. So like if you have a fellow researcher or a junior researcher, you want to say this is a good example of a sclerotic glomeruli or something, you can give that, and you need to make sure that that's something you can just give them without worrying about how that gets used. Obviously, if you're working with another company or with collaborators, you need to share that data, and by having it anonymized, you reduce the challenges of going through an IRB and all the work that goes with that and making sure the data is kept secure. And of course, if you publish results, you often have to publish a data set with your results so that you can actually let other people validate them and do due diligence on that. While we want to remove protected health information and personally identifiable information from images, we do also want to keep clinically useful information. And I'll be touching on that a bunch as we go forward. But this is things like you might need to know the age of the patient, or obviously things like gender and disease condition. And if that's stored as metadata inside your image, you want to keep it. But at the same point, you need to remove anything identifiable. And depending on how rare disease conditions are, it's easier or harder to do that. And so you can actually have to make judgment calls about exactly what gets kept and what gets removed. And our tools allow you to do that, and this is something that's a per project basis. So we'll just start with a poll. Where and why do you need to share your data? And so we'll open up a poll for this, and just curious what people actually want to do with their data. So if anyone who's on cares to do this, please contribute. Aashish, are you seeing those come in?
Yes, I do.
Okay. I want to give a little commentary as that occurs, because actually you've got that view and I don't.
Yeah, it's just looks like we just got all the entries in now. So for the poll one, most of the data is coming from research collaborations.
Yeah, so okay, we have research collaborations, some publications, some validation. Great. That's not surprising. Of course, collaboration is the heart of science. And so we'll go on and keep going about how we can accomplish this. And so what we have is the problem here is that PHI, protected health information, and personally identifiable information can appear in all sorts of places. It can be in the label images on our slides, it can be in the macro images, and we'll give some examples of those later. It can be in internal metadata, places where you'd expect it to be, like image description could have a user's patient name in it, but also places where it's not expected. Facility information shows up in surprising locations, like IP addresses of your hospital showing up in the field of some scanner data. Dates are an interesting thing because how much is too much? We need to know patient's age to help understand diseases, but you don't want dates of birth, and in some cases ages might be too precise as well, especially if you have like pediatric cancers or something. The very age of a person might actually give away who that person is if you know both that and the hospital came from. And then we have cases where people have written on images. You have a pathologist who's written like the word Gleason and a score on a slide, and hey, we all as researchers know that's a Gleason score and not a name of a patient, or that's the likely way to imagine it. But if it has some other word, you have to actually make a judgment call and say, is this PHI, or is this just a word that the researcher who was doing this has included? And the other thing is, especially with collaboration and research, we end up needing to do this for a large number of images. Doing this on 1 or 2 images is not that much work. Doing it by hand, doing it with certain limited tools. But as we get to larger scales and use more and more images, we need to make this so it's automated and only has us contribute when there's something different or unusual. So the software we have a piece of software called ImageDePHI. This is an executable desktop program, but it runs with a browser as a web interface. It's totally local. Obviously, if you're de-identifying slides, you need to keep the data in your system. You don't want to go out to a web service because you have to trust that web service would make sure it never has a leak or anything like that. So while this runs with a web interface, it is totally local. It can be done either with a simple GUI, for which we have an example on the right here, or it can be done with command line usage. So you can say, hey, I'd like to run it on this folder of images. It's scriptable, it's well behaved, so you can actually put it into your workflows. By default, it handles any TIFF or SBS or slide modality DICOM, and I'll go into image formats in quite a bit more later. It has customizable rules for those exact reasons I mentioned about dates. We might care about how it's done, or for instance, the macro and label images that you can see here. Incidentally, these are all either samples from mouse tissue with completely fake metadata, or examples from the TCGA, which have already been anonymized. So everything you see here is already, anonymized, and any data that looks like PHI here is totally synthetic to make sure it'd be bad form for us to leak PHI while giving a talk about how to not leak PHI. When we run the software, it outputs a report about what it actually did. So we took source images, which might have these long names, and outputs it to an anonymized name that is more generic to your study. And we can also store images that we failed to redact for some reason as well. And those reports are not necessary. They're just feedbacks. You can see what your software has done, and so you can see what has occurred. Those reports are really just for the person running the identification tool. Later, we could use those if we ever need to get back to the original images, but there's other ways that are more secure than that. So we'll just start with image formats. So what whole slide image formats do you need to work with? What have you worked with? And where is the your data coming from? So we'll just ask what people are using.
Okay, David, looks like we got all the polls in. So
give it a little more time in case anyone else wants to contribute here.
Yeah, looks like DICOM, Leica, and then TIFFs are the most popular formats.
Well,
all three.
That's curious. We have a very even race here between TIFF and DICOM. So those are obviously the big formats here, and SBS files are, I would have guessed would be the most common, and so it actually surprises me that they're not the leader of that. So we work out of the box with any TIFF or slide modality, whole slide image, DICOM, and we've made other software for another project where we've actually redacted Hamamatsu NDPI files, that's the nano zoomer files, and also Philips iSyntax. Philips iSyntax has the requires that you have things licensed in order to be able to work with those files. And if you end up with other formats besides TIFF and DICOM, there are literally hundreds of formats. We've integrated with open slide readers, a variety of readers, so we can read a lot of different formats. We have our own libraries for doing this. If there's a format that isn't TIFF or DICOM, we can convert it to TIFF or DICOM. So this is something where it could be that converting it's acceptable, but converting it does change data to some degree. It ideally doesn't change your image data in any manner, but you might lose metadata that you cared about. And so that conversion is not always the right choice. If you had to convert, if you had to redact a Nikon or CZI file, we certainly could develop techniques for doing that. And all the images I'm showing today happen to be bright field images of hematoxylin and eosin stains, but we work with any images regardless of the image modality. So fluorescence microscopy that could have multiple channels, like different protein expression channels, or you could have Z-stacks or time series, and our software works with those. So this is something where it's not just the common H&E values, but if you had other slides, we can work with that as well. And I mentioned that some of this is to do it at scale. So we have automation and validation at scale. If you do this one image at a time, you can check through a few images and be pretty confident you did this correctly. But if you have hundreds or thousands of images, this is not really something that you can want to be doing with your time. You'd rather be doing your research. People are not very good at being consistent. If you break down the date, you might write it month, day, year, or they might write it year, month, day. It might be context dependent. If you look at how you've named all the files in your documents folder, I bet you weren't particularly consistent at them. Most people aren't. And so we want this to be consistent. We want to be certain that every time we de-identify images, we've done it correctly and thoroughly. And so tools need to detect novel or unexpected data. We need to approve things that we know are good, not deny things that we know are bad. So this is a whitelist is a good thing, a blacklist is a bad thing, because if a blacklist is the only thing we're using, if something's not on that list and is wrong, we will end up letting it through. And some of this is things like letting have data for, say, an image property like microns per pixel. You know, we expect a number to be in that field. And if we read that field from an image and it had a text value, we might say that's surprising. Now, it might be that someone has just written out, 0.5 microns per pixel and added the text there, but it might be that there's something odd going on. And so this is where our tools are designed to validate these values. So this value must be a number, or this value must be a date, and therefore I know how to deal with it. If not, stop. And again, we do want to keep the clinically useful data. We want to keep label and macro images if they're good. And there's reasons why we want to do that. And so as an example, we have, to support this, we have redaction profiles. So at the minimum, you could say, I want to keep primary image data and nothing else. The image still has to work as an image. I can still run it through my algorithms, but I strip everything off of it. This is the absolute safest way. You end up with an image that has nothing but actual imagery of the primary image. You don't have any macro, you don't have any label images, but you also lose metadata, possibly like the objective lens used, or the actual properties about like exposure times, or maybe a particular imaging modality that was used. We can have things be strict, but not quite that minimal. We can keep metadata that can't possibly be related to a person or facility, the crop zone that was on the slide, things like that. We can anonymize dates, keeping only years, for instance. It's a popular choice, but we can also be based on rules, say, I'm going to round people's ages to every 5 years, or whatever your policy is for this particular project. And then label and macro images is something we have to check. And in some instances, we have to remove because there's nothing else we can do with them. So as an example, we have here a slide in the upper right that has an area of tissue. It shows the scanned area with that little box around it. And this is useful to see because, for instance, I can very quickly see that's not a needle biopsy. It's not. So I can tell what sort of biopsy it is just by looking at it. And so this has virtue. And I can see, oh, this is the overall scale. I know my slide was about 25 millimeters tall. Therefore, the tissue is a particular scale. All of that gives context. But the problem here is that in this case, the label extended down into the area within the macro image. And if I brighten it, you can see that I can actually see a barcode in that label. And if I use a barcode reader, I can actually read it. Now, in this case, it's just a block number. It's not interesting. It's totally safe. But if it wasn't, I would have to redact that in some manner. I could just throw out the macro image, but then I lose the macro image. I could develop a custom rule and add it to the system and say, hey, I just want to remove part of the macro image. So this is something where if your lab's consistently overlapping your labels into the macro images, maybe you need to talk to them about a process. Maybe you just need to throw out the macro images, or maybe you know that it's got only barcodes, which are totally safe, and you can let them through. But you have to decide that on a sort of a batch case basis. The other thing is we have label images. This is a synthetic label. Totally artificial name, totally artificial date of birth, totally artificial barcode, block number to list where it came from. And but if this was a real label, I have to remove this. Clearly, it's got a patient's name on it. And so there's some software that requires a label image to exist. So we could either remove the label image entirely by throwing it out, or replace it with a black image. And we've had people say, hey, I'd like to replace it rather than with the black image with a custom image that has my own internal identifier on it. That can be done too. And so these are all options about what can be done. If you need to have a custom image, we could certainly help you integrate that into the software, but that's not going to be done by default because we don't know what you'd want as a custom image. And so this is where we have these redaction profiles and say, how do I want to redact it? Let me set that up for this batch of data, for this project. This is the redaction method I want. You have to validate things. So our software is meticulous about this. And so this slide tells a story. I've simplified it to be as small as possible to show this off. Each of these is a hex dump, hexadecimal representation with the text data that's within an SVS file. So these are all actually, SVS is a type of TIFF, and the TIFF file contains a set of tags. The tag can say, I've got an image description. And then it has those tags stored some way. Then it's got image data, possibly more tags. It goes in different orders. There's a lot of variations in TIFFs. Could write a whole thesis on the whole topic, but fundamentally I had a slide where I had a image description where I started with a message. I wrote public message on it, and then I ran it through someone's software. I won't mention who, not ours, and I changed that value to the word secret value. Clearly not an appropriate value if it was actually PHI, but this is just a proxy to say, what if I were to do this? And then I saved it. And the first thing I noticed was in this original file, which is on my upper right, it has both that original message that I had and the new value I typed in. It means that that software probably is doing something like a tool that is public and common that we can talk about. So if you're dealing with TIFFs and SVS files, you could use a common library called libtiff. This is installable on Windows, on Linux, on Mac OS. It's just a command line tool that gives you a bunch of tools for working with TIFFs. One of the tools is called TIFF set. It says, I want to replace the tag image description with a different value. And so on my left, I have an example of what this did. I said, I want to replace image description with redacted value. And then I did a hex dump of my file. And now I actually have 3 values in my file, not just 2. I really should only have 1. The first type public message, I used this external tool, added secret value, should have gone rid of public message, it's still there. I then used TIFF set. I said, let me change that to redacted value. And now I've got this 3rd value there. TIFFs internally have a pointer that says I've got image data, I've got an image description, I've got other things. Internally, they point to those values. And what this software did is it wrote the value, the new value at the end of the software, at the end of the file, changed the pointer to say point to that new value, and then just don't point to the old value. And so if you looked at this with a variety of tools, you would only see the redacted value, but it's still in your file. And so this is the problem, because you really don't want to leave that data in the file, because when you share this as part of your research project and someone looks at it, they go, hey, I can do some sort of search through this binary data and find the original redacted data that you didn't want to keep there. So we are meticulous that when we run it through ImageDePHI, we rewrite all files. This is slower than just doing this modification. It's very quick to just add a few bytes at the end of a file and change a pointer. It's much slower to write a file, especially since a lot of our whole slide images are gigabyte or more in size. And so we have to do it. This is not really an option. If your software is super quick at doing this, it's probably not doing the job perfectly correctly. Now, you can speed this up by having good storage, but the point of this is that you need to make sure your tool is validated for actually removing the PHI, not just hiding it. And so we've done the hex dump on our file here to show that we indeed have thrown out removed the data, and actually we just wrote a brand new file, so it's not modifying it, it's rewriting it. And anything that we remove, like we remove a macro image, we don't just stop pointing to it, we actually remove it. So all of those other things in the other software might be left behind. Software might do the right job, it might not. You need to validate whatever software you use to make sure it's doing the correct thing. And then just as a reminder, if anyone has questions, feel free to enter them in and ask as we go. Otherwise, we'll get to a live demo. So I'll give you a little bit of time to say if anyone has any questions here.
This is great, David. Yeah, so as David said, any questions you can type in, and then I can announce it here as well. Or you can do it after the demo as well.
Okay. So I've got the software running locally. I have just typed a command line, so ImageDePHI GUI. It opens up a web browser, but it's just to my local port. So this is the local system. I can choose what port, but that's not important. The simplest case here is I really have only 3 things I'm choosing. I'm choosing a directory full of images, I'm choosing where I want to redact them to, and what rule set I want. By default, there's a default rule set, which is not quite the minimal. It's a fairly strict, but not minimalist. And if I select this, so I'm just going to say, hey, I've got a sample set here. And when I select that, it's going to load that sample set. It's parsing through the files. This is relatively small. There's like 27 files in this directory. And you can see it came up tolerably quick. This is on a nice, fast NVMe drive. Each each file is listed with an image. This is the actual whole slide image at a reduced resolution, obviously. We have label and macro images if they're present, not if they're not. We have a little checkbox that says, can I redact this successfully? Do my rules cover everything I know that's in here? And that's just a simple yes, no, but I'll show an example when it's no. And then it's showing things that automatically will redact. So we've said, hey, I've got some sort of digital slide ID. In some instances, this is a serial number. Some instances, this can be an IP address. In general, we feel that's facilities information, so our default is to redact it. We have a date, we're just getting rid of that. We might want to keep that, but here we're just getting rid of it. And then we have file names, and we don't want to keep, this is actually not the file name, so the file name is what's on the left. This is a piece of metadata inside the file called file name. These are confusing things because you can also have an extra image inside the image called thumbnail, which is a image that may or may not be an actual thumbnail of the main image. It's just another image with that name. And so by default, we don't want to keep whatever file name it had internally. That could be something that's exposing information that we don't want. By default, we can get rid of it. And similarly, we have image IDs, which look a lot like those file names. And we can keep scrolling, and there's other things like this scan scope ID. Hey, this was scanned with an Apereo scan scope. There's a couple things that may or may not matter. We probably don't care about time zone. This user, which is all zeros, clearly that's not interesting. But these, maybe that's the scanner user. We don't know. And then there's things we really do want to keep. We want to keep the application, the magnification. So this was used with a 20x objective lens, exposures, things like that. And then there's things that we must keep, like the bits per sample. That's actually part of the image, and we have to keep that. And so it just keeps going on and on. And there's some things that we really want to keep that are long and complicated. Like, for instance, there's something called an ICC color profile. This is when you make a scan, the scanner should be color calibrated so that when you view it on your monitor, which is also color calibrated, it looks correct. Hopefully your software deals with all of that correctly. I've got a standard monitor in front of me that uses the sRGB color space, and so when I look at images, they're rendered in the sRGB color space. But I could have a P3 monitor or whatever, and this ICC color profile is vital to rendering that image correctly on whatever your monitor is. So we know we can't get rid of some of this stuff. You could get rid of things like this, left field, where this is like where in the image, probably in millimeters or something, that it scanned it. I don't know what that value is. And so we have a whole variety of values here. And microns per pixel, I happen to know that's what this means. This is critical for scale, especially if you're going to run machine learning on it or something like that. So all of this just shows our default. I can pick an output directory. Where do I want to write this? I can just say, hey, stick that in my results folder. And if I don't say anything else, I can just say, hey, redact it. Now, again, I have a nice fast NVMe drive, so this is going to go pretty quickly. These are comparatively small images, ½ gigabyte or so each. And you'll see some are slower than others because those are bigger images. And this is going through and tolerably quick. And that's because I have a fast drive, and these are not the biggest images in the world. And when it finishes redacting, it comes up with what it's done. And you can say, okay, my labels have been blacked out. I've gotten rid of the macro images. Some of those values which you saw earlier are no longer there. It tells you I wrote them out, and you can see that it still has an ICC color profile, but there's other values just gone. It no longer even lists them because they're not present. And that's great. That's precisely what we want if we wanted to get rid of those values. If instead I picked something that had some missing values, for instance, if I pick this directory, I've got 2 images here which have values that I don't have rules for. And again, if you have questions, please type them in, and we'll get to them as they go.
David, just to interrupt you, looks like you lost your video stream, but we do see the demo right now.
Okay, I'll just proceed. No, I'm having trouble hearing Aashish here. So here I've got some images that don't have certain rules. So like this is saying it doesn't have a rule of what to do with lens info. If I click redact here, it will show that it has missing rules. If I select continue, what's going to happen is it's going to redact the files it can, and then put the others in. It'll make a copy of the others in a file in an area saying I failed to redact these. So if I continue with this, instead of having my 26 or whatever images I have here, I'll get 24 redacted files and 2 that didn't. This is much safer than keeping these images that you need to do, and you can go back and look at them and go, what happened here? So if you have this rule set, you can say, hey, stop, just don't even process it if I can't do it. Otherwise, you can say, hey, this is okay. I need to keep the ones I can, but not the ones I can't. I'll deal with those later. So you have those as an option. Instead, I could change the rule set. So I could say, hey, I'd like to choose my examples. I'd like to choose rules and say, I'd like the modify date rule set, or that's not the one I want. I want to go to the minimum rule set, for instance, which is get rid of everything that I don't know anything about. And I can redact these. Now, those unknown tags are just going to be gone. So those just go away. And so if I run this, it's going to then get rid of these, and we'll basically lose both the label and the macros in this case. Well, that's okay because I get to keep all my images. So, or I need to tune the rules. And the rules are just a custom YAML file that has specifications. They're somewhat hard and annoying to make up, but we can do that, and we can work with it that way. And so again, if I wanted to choose something different, I could go back to, for instance, my original set here and say, hey, I'd like to use my date rule set, and say, I'd like to use the modified dates. So instead of getting rid of dates, it would actually keep my dates and move them to round them to the nearest year. So somewhere in this column, I'll have the dates that are being modified. Anyway, so it's showing the dates are not going to be removed, but they will be modified. And so you have these options. So this is fairly basic. We basically choosing an input, an output, and this rule set. The complexity is making sure the rule set does the right thing, and that is basically the interesting aspect of this. So I'm just going to end the live demo because we've shown off what that does, and then go back to our slideshow here. Actually, I'll leave it on here. Well, Aashish, can you see if there's any questions?
There's one question, David, if you can hear me. If you cannot hear me, then I posted that question to you on the chat.
So we've had 2 questions. So basically, we are using for opening and reading images, we are using Python libraries depending on the image format. So under the hood, there's some Python code here. We're using TIFF tools for reading TIFF files and PI-DICOM for DICOM files. We are not actually decompressing images. So we actually read a specific portion of it so we can decode it to show it, but we never recompress the image in this case. We basically are just taking the bits of the image and moving them to guarantee that we're not introducing any artifacts. And so that is, so we validate things with tools like OpenSlide. We have our own library like Large Image, which can read hundreds of formats, which OpenSlide is one of the readers for that. But we actually don't decompress these. We are literally using libTIFF, the very one that I said the TIFF set worked poorly on, to read the actual portion of the image from a TIFF using PI-DICOM for reading a portion of the DICOM, and then various like libJPEG and OpenJPEG to read various encodings in that. And then the other question we had was that we had earlier showed the file where we had those extra image descriptions, and our software definitely would get rid of. So the question for people who can't see it is, someone asked if with the PHI that we showed on the earlier with the hex dumps, when we rewrite those files, regardless of how many times another piece of software wrote it badly, because we completely rewrite the file, whether we're doing TIFF or DICOM, we rewrite the file and all of its parts, and we only put in the data we want. So in this case, we don't decode the image. We say, here's the image data, copy it into the new file. Here's the metadata. The metadata we completely rewrite. And this is with in this case for TIFF, we're using the Python library called TIFF tools, and that writes that data and is very pedantically correct. This is guaranteed to be a correct TIFF file that meets all these criteria. You'll see TIFF files with all sorts of minor petty issues that they're still readable, but they're not technically conformant. We're guaranteeing that we write conformant files, whether they're SBS or DICOM or TIFF. And so going back to our slides. So we'll add another poll question here. How many images do you handle in a typical project? So what are people using out there?
Okay, David. It looks like we're getting thousands as the top choice, followed by 10 to 100 as the second one. Looks like they still cannot hear me, but hopefully you all can hear me. So thousands is the looks like the top choice.
Well, yep. So thousands, that's one of the things. Like if you're doing thousands, you certainly don't want to look at every image directly with that. Yeah. So this is why integration is important. So we have a graphical interface, which you saw, and this is great for testing rules on small batches. If you have, 50 images, it's fine. If you literally are sticking 1,000 images in it, you're going to be scrolling a lot. And so what you want to do is you want to have representative images. And if your lab's producing the images directly, any given batch is likely to be fairly similar. But data that you got from 2 years ago might not be the same as data because labs techniques change and things go on that way. So the program is designed to be batch capable command line, so you can do this at scale. You can do this either just from a command line prompt to try it out, or it's intended to be scripted in this sort of a Linux philosophy way, though it works just fine in Windows and OS X. And you can have any number of rule sets. So the different policies depending on use and sources. So you may know that this set of data, you need to remove the macro images, but this set you can keep them. Or you might say for this particular case, we want to do something different than on this. And so that integration is critical. And then, as I mentioned, we do write out this manifest file when we do the redaction, and the command line does produce logs to different verbosities as you need it. And that's fine, and you end up with that. But there's an important thing, is that when you've shared your data, you have this, a fellow researcher may come back to you and say, I have a really interesting case here. You gave me this image and it's fascinating. Can we learn more about this their patient? In this case, you might need to go back to where there actually is PHI so you can look at more information. And so this has the concept of an honest broker. So if your research project needs more information about a sample, how do you get back to the original image? If we've redacted it correctly, there shouldn't be anything else. And as part of this, we don't add extra identifiers to the redacted image. We basically have this where we looking at it and can see that the image does not contain any information that we didn't put in ourselves. And so we aren't adding an ID, we aren't adding things. So how do we get back to it? We can look at the image itself as a basis of comparison. By default, we don't recompress the image. So we can actually make a hash of those bytes and say, hey, this image is the same as the image that we copied. The bytes are the same. The metadata may have changed, but the bytes of the image are the same. So that's a way we can do this. And we can do this fairly efficiently. We can do it fairly quickly. You have to have access to the original image. That's still at the organization that did the redaction. You could theoretically save this in a database to be faster, but then that database is something you absolutely must keep secure. That's got to be inside your protected zone that has PHI in it, or has data that lets you get to PHI in it. So that has to be protected, and that would be if you want to do that, because you need to do this often. The other thing is that if you did recompress an image or cropped it, things like that, there are still ways to get back from the image basis as a comparison. They're more work, they're slower, but they're totally functional. And so we can help with all of this. So while we have this software, ImageDePHI can be freely used. It's open source. You can download it, you can modify it, you can use it. But rule sets can be tedious to develop. If you find this challenging, or you just don't want to do it because you want to do other things, we can help with that. This is a service we can provide. You might need additional options to keep or anonymize some data. So if you have something you want to keep or inject, you go, hey, I want to add these tumor reference codes that are from my reference database to this data so that when I pass it into my machine learning pipeline, it knows what to do with it. This is something that can certainly be done. We've done this for other projects where we've said, hey, we want to get data from a specific database using actual PHI, look up in the database, get tumor information, add it to images, have the tumor information in the image, but not the PHI in the image. And so these are the sorts of things that you can have as customizations and readily add into your system, so that way when you're doing your learning or machine processing or anything else that's with it, or even if you're just sharing it, that metadata is with it instead of having a sidecar file with all that information. We can do this as a service. We can decide the basic policy, the anonymization level you need, how we're dealing with dates, what we're getting rid of, what we're keeping. We can run this on representative images, get a report of tags that exist. Where does this data exist and what are you going to do with it? We can tune the rules and add special cases, and then we can run this on your actual data. If there's anything we missed, it'll come up and say, hey, we missed this tag. What do you want to do with it? This tag's surprising. It contains information we didn't expect. And then we iterate this as needed as new data occurs. And so Kitwork can be your partner moving forward. You can try this out. We've got a link here. We'll share the slides. We'll share the links. If you need help, you can just ask a question on our Histomics forum. We can also add paid support or training. We can do a lot more than that. If it's a short question, we're happy to help publicly. As the questions get longer, we are a business. We need to do this. And then if you are doing a research project and you think, hey, we just like to have you guys be a subcontractor on a research project to help us with this or with any histology work, we're available for that. And we are available for software customization, rule expansion, new features, custom workflows. All of that can be integrated. How do we do it into your lab to make it work for you? How do we integrate is all something we can work with. Related tools we've mentioned, HistomicsDK, while its name internally means the toolkit that does some basic image processing, it's also used as a catch-all for Histomics platforms in general. And Digital Slide Archive is a reference implementation that we have a method where you can install those tools directly on your desktop, or also can be installed in the cloud. And then if you need to reach us, we can be contacted fairly easily, and we go from there. Any other questions?
Thank you, David. This is great. Any questions from our audience here?
Thank you, everyone, for your time.
Yeah, thanks all. Have a good afternoon.